Hello to all dear students. So today onwards I am going to start a series of lecture on antiviral drugs. So first of all we'll study how the viruses enters inside the human cell and how it actually multiplies so that we can know the target by which we can inhibit the viral multiplication. So suppose this is a human cell and this is a virus. Viruses, these are obligate parasites. And these viruses, they contain either RNA or DNA as the genetic material. Apart from this, in some viruses, these genetic materials are covered by a coating known as nucleocapsid. In some viruses, nucleocapsids are not present. They are free. Genetic material are free, and in some viruses, genetic material are covered by this nucleocapsid. So, what exactly happens is the first step is the virus they fuses with the human cell. After fusion, it will send its genetic material inside the cell along with the covering. So, along with nucleocapsid, the genetic material comes inside. If nucleocapsid is not present, directly the genetic material comes inside. So after the genetic material has come inside, the genetic material is made free of this coating, the nucleocapsid, and this step is known as uncoating. Now, if the genetic material is DNA, it will directly start working. But if the genetic material is RNA, then it is converted into DNA. Like in case of HIV, there is a reverse transcription. RNA is converted into DNA. So this RNA, particularly in HIV, it can be converted to DNA. Then this DNA, it will start multiplication. Okay, many molecules of DNA will be produced. Replication will occur. So several molecules of DNA will be produced, and now these DNA, it will result in the formation of a protein. Okay, so replication occurs, transcription occurs, translation occurs, and protein synthesis occurs, and because of which these proteins are being produced. But the problem is these proteins produced, they are inactive, they are not active. So the next step is to make these proteins active. Okay, so these inactive proteins are being converted into active proteins, and when all the proteins are activated, then complete virus is generated inside the cell. Now now after completion of this complete viral generation, so now this virus, they will be released from the cell and it will start infecting other cells. Okay, so this is the general method by which virus multiplies. So we have targets for inhibiting these viruses, like we have got various steps by which at which we can inhibit these viruses. So we shall check out the various steps by which we can inhibit this viral multiplication. So very first step is, right in the beginning, the fusion can be inhibited. Okay, so if viruses do not fuses with the human cell, it cannot multiply. So fusion can be inhibited. So we have got group of drugs known as fusion inhibitors. Next step was, after fusion occurs, the viruses, they will send their genetic material along with the nucleocapsid, and the nucleocapsid will be dissolved. Okay, so we can inhibit this uncoating process. Okay, so we have got group of drugs known as uncoating inhibitors by which uncoating can be inhibited. So drugs like amantadine, they act by inhibiting this uncoating thing. Next, the third step for this viral multiplication was, once uncoating occurs, next what happens, if the virus are RNA virus, it has been converted into DNA, isn't it? So we can inhibit this step also. So we can inhibit reverse transcription. So this is the another step. Fourth option by which we can inhibit viruses are, the next step was, once RNA has been converted into DNA, next what happens? Replication. So many DNA were being produced, isn't it? So our next way by which we can inhibit viral multiplication is by inhibiting this replication of DNA. Okay, so we should inhibit replication. Next option by which we can inhibit viral multiplication was, this DNA is so formed. this DNA will start producing, means they will undergo transcription and then translation, and they will produce the protein which is usually inactive protein, and these inactive proteins are being converted into active protein, and this is done by a enzyme known as protease. So this protease enzyme, it will convert inactive protein into active protein. So we have got group of drugs that will inhibit this protease enzyme known as protease inhibitors. So what this protease inhibitors will do, it will inhibit the enzyme protease, because of which active proteins are not formed. Next, the sixth step was this active protein so formed, they are being converted into the mature viruses. So we have got drugs that can inhibit maturation of these viruses. Okay, so we have got maturation inhibitors. Lastly we can inhibit the release of the viruses from the cell, and in this is mainly done by a new drug known as oseltamivir. So these are oseltamivir group of drugs. The drugs which is mainly used for treating swine flu and bird flu. So they mainly inhibit the release of viruses from the cell. Okay, so these are the drugs that will inhibit the release. Okay, so these are the general mechanism by which viruses can multiply. Okay, so next we shall now start discussing regarding viruses one by one, and in my next video I'll be discussing about the human immunodeficiency virus, that is HIV virus, and then we shall see how we can treat these viruses. Okay.